Hi guys, we are back again, and today we are going to talk about the cell membrane. The cell membrane or the plasma membrane, we've already talked about this, is selectively permeable. Why is it selectively permeable? We'll talk about it later. It is made up of means two layers of phospholipids. Here are the two layers, 1 and 2. Phospholipid, it has a phosphate head and it has these hydrocarbon tails. The heads, this is the hydrophilic part and the inner part is the hydrophobic part. So here is the water loving, the outer part, the inner part is the water hating. And these form two membranes. In these two membranes, there are a bunch of membrane proteins. And it is via these membrane proteins that particles are going to pass through. And that's why it's called selectively permeable. That means it will only allow certain particles to pass through. These proteins are freely floating. And these are randomly placed. And that is why the model to explain the structure of the cell membrane is also called fluid mosaic model. In the phospholipid heads, you will see a bunch of glycoproteins. We'll talk about this again. You should also see cholesterol, which actually in the cell membrane, which actually limits the fluidity of the cell membrane. So anyway, let's go into more details now. There are a bunch of proteins, as I told you, in the cell membrane, and we are going to talk about all these proteins in the next slide. The first one is the channel protein. The channel protein goes all the way through and has a pipe, a channel, a tube through which particles are going to pass through. Of course, they are going to be selective based upon size. For example, this particle cannot pass through because it's too big. Or on the shape, this particle cannot pass through either. The shape is not suitable. So based on the particles that are correct size and shape are going to pass through these channel proteins. The next one are the carrier proteins. Carrier proteins are very, very selective. So carrier proteins are like channel proteins, but they are selective. In other words, they are going to bind with very specific molecules, very, very specific molecules. And they are not only going to accept it, but they are going to move the particle through them like the channel proteins. At the same time, they're changing shape. Look at this carrier protein. It's changing shape as it is passing the particle. Yes, carrier protein is slower than channel protein, but it is more specific, more controlled transport can take place. Cell recognition protein, I told you that the glycolipids are attached to these chains, glycoprotein chains. And these chains are the ones that are going to give identity, a recognition to the cell. So just like all the members in the family have a particular smell, and the dog in the family can recognize, oh, I belong to this household with these members, and say, for example, I walk into that household because I don't have that smell. I am a foreigner. Same thing. These glycoproteins will help the body differentiate between self, and non-self. So all your cells have a certain self-recognition glycoprotein attached to their cell membrane, because of which the soldiers in our body, our immune system, do not attack our own cells. They will attack any cells which have non-self, which have different glycoprotein. This is how immune system attacks pathogens. Should not attack our own body. Of course, there are autoimmune disorders. That's a whole different ball game. We'll talk about it later. Receptor protein. Receptor protein receive certain molecules, special molecules. As soon as they receive the molecule, note they will change shape. So they will receive, and at the same time, they will change shape. So they will change shape, and once they change the shape, they are going to set up a signal. And this signal is going to, as you can see, it is changing shape, it is becoming a dimer, it is setting up a signal, and then this allows an inactive protein to become an active protein. All right? Enzymatic protein can be called as biocatalyst. Why biocatalyst? Well, bio, you understand, it has to do with living. Catalysts are anything that changes the rate or speed of reaction. Rx is reaction. So here what happens is an enzyme. The enzyme has this site or groove which is called the active site, into which this substrate is going to bind. This substrate is nothing but a reactant. Here's my enzyme. Maltose is binding onto this groove of the enzyme, which is called the active site. And once it binds, you notice that the maltose is going to break down into glucose. All that the biocatalyst is doing is going to change the rate of reaction. It is not going to change the reaction. It is, so if A plus B is giving you C, it is still going to give you C, but now it is just going to change the speed. Either it will increase the speed, that means it will make it go faster, or it will decrease the speed, that means it slow down this reaction. This is the enzymatic protein. Junction proteins are those that bind, attach, connect two different cells. The first one we are doing are the tight junctions. Tight junctions will are like hooks, like a zipper-like fashion they will go. As a result, these two cell membranes, these two cells are now what happens is are hooked together very, very close. This will not allow anything to seep through. For example, tight junctions we can see lining our digestive tract, example our stomach. So because of these tight junctions, the digestive juices in here are not going to seep down through and destroy the other parts of our abdomen. The other one is gap junction. Gap junction is there are cells, but now these cells, the cell membranes are going to fuse, and as a result there's a pipe or a tube. So here not only, so I'm going to make it like this, a little different from here, okay? So let's make it like this. So here is a pipe or a tube. So now what happens, not only these two cells are closely connected, identical cells are connected, but now quickly material from here can pass through from first cell to the second cell, typically seen in the cardiac muscle cells. So each cardiac muscle cell is connected, as a result of which, because of these gap junctions, as a result of which the stimulation can spread through, and the whole heart can now what happens is contract in a rhythm. All the heart cells are going to contract in a rhythm. So 1, 2, 3. The malfunctioning plasma membrane proteins, there are 3 that your book has discussed, is diabetes 2. In diabetes 2, here again there is, like I said, here is a receptor. So insulin comes here, and insulin is going to bind to these receptors, which is going to set up a signal. So as soon as it binds, it is going to become active. Let's make it active. Let's make it a crinkly line. So it's going to be active. As soon as it becomes active, now what happens that the glucose can be pushed into the cell. Now in diabetes 2, insulin is there. But guess what? Because of overabuse, these receptor proteins are not going to function. We don't have these receptor proteins. So as a result, insulin is there, but nobody is binding the insulin, setting up a signal so that glucose can enter the cell. So basically the cell starves to death. There's excess of glucose in the blood, and the glucose comes out in your urine at a very, very high level. Color blindness. Now you see color because of special cells, and these are called cones in your eyes. The cones, you see there are 3 cones, blue, red, and green. So the typical color blindness I'm talking about is the red and the green color blindness, which is typically, it's a genetic disorder and seen in males, typically seen in males. Now here what happens is that when the light of a particular wavelength comes and hits a cone, the photopigment inside this cone will get excited, and the sodium proteins, the sodium channels, these sodium channel proteins, or you can just say channels, these channels will close. And so what happens is that action potential, well, I guess y'all don't understand the word action potential. Well, a signal is set up and you can see the light. All right. But because you have faulty sodium channels, so what happens is that even when the light is coming, the photopigment is missing. You don't have the photopigment green and red, so no signal is set up. So sodium channels, now what happens is remain open, do not close, signal is not set up, you do not see the light. You do not see the color. You see light, but you don't see color. So you don't see red, you don't see green. In cystic fibrosis, there are faulty chloride channels. So here what happens is that because of these faulty chloride channels, the sweat as well as any secretions are going to be very, very thick. So your sweat is going to be very, very thick. Your mucus in your lungs is going to be very, very thick. The pancreatic secretion is going to be very, very thick. And basically, these people, if their mucus is not removed, they are going to die because, gas exchange cannot take place. So in cystic fibrosis, there is faulty chloride channels. In color blindness, it has to do with sodium channels that do not close because the photopigment in the cone cells is missing. And in diabetes too, again, it has to do with these receptor proteins. So with this, we come to the end of plasma membrane proteins. If there are any questions or concerns, please contact me. Thank you. Have a good day.